Okay, well, welcome back and thank you very much. My name's Alta Sharo. I am a professor emerita of law and bioethics at the University of Wisconsin-Madison and would like to speak to you today about some of the ethical challenges in this area. I'm going to start first by sharing my screen and to mention that I do have the following conflicts and biases: a lot of committee work with the national academies and with the International Society for Stem Cell Research on both stem cell research and also human genome editing, and consulting for the companies listed here at the bottom. None of the consulting directly on point for this particular meeting. Many of the questions that need to be discussed in this area are not entirely new. They've already come up in the context of prenatal interventions involving surgery or other kinds of therapies. So questions about, how many patients you have, who gives consent. These are things that we've already encountered, but in some cases, there are special risks and opportunities that are being presented by virtue of the fact that it's gene therapy and genome editing. And the politics around abortion to some extent is also going to be affecting the calculus in these cases. When you talk about who the patients are, again, some of these things very familiar because we're talking about a fetus and a future child. By definition, there's no ability for the person who's likely to be most affected to exercise any choice. It also raises the interesting point that some conditions may, over time, because of your new interventions, affect what is considered to be viability. And that, for the moment, is a key marker from a legal point of view between the moment at which a woman's own interests are predominant and the moment post-viability when a state's interests in the fetus can become predominant and can limit a woman's choices quite significantly. Obviously, in theory, there is the concern about liability claims against the physicians for negligence, and in theory, even for the parents, if they went ahead and decided to have a child with severe disabilities. The courts have been very unfriendly to these claims, particularly to those against the parents. But it does begin to raise, especially in this political climate, the question of whether or not such claims may have more strength when they come from a choice to intervene versus a choice not to intervene in utero. The ability to offer people a choice that they can then say no to can change how it is that we think about responsibility in these contexts. From the point of view of the pregnant woman, we are looking at a situation in which, in general, the woman herself is healthy. She's not the patient. And therefore, just as in vaccine trials, whenever you're going to impose any risk on somebody who's currently healthy, you tend to be far more cautious. And that is certainly going to be part of the discussion in terms of benefit and risk. I'll note, by the way, that I'm talking about the pregnant women. I do appreciate that in some circumstances, we might be talking about a non-cis female capable of pregnancy, but for the moment, that is not a common situation. I want to take a moment, though, to really focus a bit on the way in which we are now seeing a kind of a rise again of social and legal pressure on women to act on behalf of their fetus or future child, even at the sacrifice of their own interests. We saw this in the 1980s and 90s with both civil and criminal efforts to get women to have cesarean sections that their physicians had recommended. It seemed to die down for a while, but it has come back, and it's coming back even more strongly because it's been tied to the abortion debate and the nature of female sacrifice on behalf of her role as a child bearer. We've seen now a growing trend in the US and in some other countries to prosecute women criminally for purportedly harming their fetus. So a woman who miscarries can be the subject of investigation to find out whether or not it was something that she brought upon the fetus by her own behaviors, and has actually been criminally prosecuted in a number of cases. There's one woman currently in jail still for this. And this can also apply to stillbirths. So we're in an atmosphere now where we need to be particularly sensitive to the way in which even the offer of an intervention can play into a larger context of notions about responsibility on the part of the pregnant woman to accept all risks to herself and even theoretically to her fetus in order to try at all costs to come out with a better outcome at birth. Um, I also would just note that there were older discussions in the past about how you manage this maternal fetal conflict. Certainly use of dangerous substances that were addictive or non-addictive was a focus. But if you go back to the 1980s, you actually see in the literature and among some practitioners active discussion about whether it was wrong to conceive a child, knowing that you had a strong genetic risk of a genetic disease of some severity, or whether it was wrong to conceive without having taken advantage of prenatal diagnostic opportunities. The result is that, again, if you begin to offer people prenatal interventions as an option, it is going to raise, again, questions about what is the degree of responsibility that we will impose on parents, and particularly on women, when it comes to trying to assure an optimal outcome for their children, as if they didn't want it already, of course. The International Society for Stem Cell Research, which has been issuing guidelines for both clinical and research context for stem cell therapies, recently updated its guidelines, and for the first time now has actively incorporated genome editing into these guidelines. And this is one of the recommendations, that the clinical research for in utero stem cell or genome editing based interventions should be undertaken only when it offers the prospect of a benefit that's greater than that of postnatal interventions. This is very tricky, obviously, if there is such a postnatal intervention, because the earliest stages of research, as we've heard, pose enormous questions. And so being able to argue that the prospect of benefit is greater is challenging. But I would also, note that it follows an interesting pattern in which we tend to be more cautious about the fetus than we are about the neonate. When if you do an intervention on a fetus and it's unsuccessful, it may in fact result in a miscarriage stillbirth, in which case the unfortunate effects are not suffered over years by a live-born child, whereas with the postnatal interventions, you're facing that. So once again, we kind of see that there's a tendency to be more protective of fetuses than of live-born children. And it's probably worth at least asking ourselves whether or not we are committed to this approach, or if it is simply an artifact of the politics around us. I'd also note, as an aside, that the ISSC guidelines also emphasize the importance of having an institutional capacity for autopsy or for follow-up, which I'm going to talk about briefly later on. The long-term follow-up of these children is something that people have already mentioned, and one person at least said that ideally it's lifelong. That's not true only for genome editing. There are other therapies that require this kind of long-term follow-up, but think about what that means. The PI is likely to retire or change. The the clinician who's most associated with the child and the parent also very well might change over time. The funding for such long-term follow-up may dry up. So these are the kind of logistical concerns that need to be anticipated when you're looking at something that may have lifetime follow-up. And particularly when in the course of that lifetime, some of these postnatal therapies might be advancing. And one question will be whether or not the prenatal therapy has precluded some of the advances that may occur later. With regard to the special attention, people have already given a lot of time to looking at the probability of the editing of gametes, male or female. I would only add that in the assessment of the risk of this kind of unintentional editing of those gametes, we might want to also incorporate some attention to whether or not these unintentional edits would tend to make those gametes incapable of fertilization. If that were true, then the risk of passing those unintended traits along has been severely reduced, and it might affect this benefit risk calculation. When it comes to the regulatory situation, interestingly, NIH-funded research or institutions that have voluntarily agreed to follow all of these research rules that the NIH has issued, including these special subparts like the one for pregnant women, these rules don't necessarily apply automatically just because you're doing something that the FDA regulates. That said, even though the regulations, the FDA don't have this section, the FDA recommends that these particular rules be satisfied in theory, they have the power to decide not to impose these rules, but for the moment they do. So in the first case, you have the prospect of direct benefit for the woman or for the fetus. And in that case, more than minimal risk to the fetus is permitted, okay? That is comparable to the kind of thing you would see postnatally. More than minimal risk is allowed to be imposed on a child if there's a prospect of direct benefit for the child, similarly here for a fetus. But if there's no such prospect of such a direct benefit, then the risk to the fetus is not allowed to be greater than minimal, which is usually defined as roughly the risk that is associated with an ordinary medical visit. Now, this poses some real dilemmas because we've already heard some discussion about phase 1 trials. Can a phase 1 trial actually hold out a prospect of direct medical benefit? Particularly if you're talking about those dosing trials that people had discussed in the previous session, if the earliest phase 1s are done with extremely low doses, then the likelihood of a direct medical benefit might be affected adversely, and it makes it difficult to say that you're not exceeding a minimal risk without a prospect of benefit. In other words, can one meet this minimal risk standard for some of these interventions? And if not, should the FDA reconsider its recommendation to comply with these particular rules? And I would also note that these rules apply even if a fetus is going to be aborted, which means that if there were a situation in which ideally, rather than using animal models, which may not be generalizable to the human, either in terms of the way in which the intervention, the systemic effects of the intervention or the dosing rules, if the animal models are insufficient, there may be situations in which ideally you would want to actually do these tests for things like dosing or initial studies on a human fetus, a developing human fetus. And if it were about to be aborted, then again, you would not have a live-born child who is going to be subjected to risks and adverse events. But these rules about no greater than minimal risk also apply even if a fetus is about to be aborted. Now, we now are in the midst of a discussion about the use of tissue from aborted fetuses, where the key element has been to make sure that the prospect of the research does not affect the decision to have the abortion. So woman decides to have an abortion, that decision was made, only then is she approached about whether the cadaveric tissue can be used for research. A similar kind of model could be applied here if there were research that was important to do in a fetus, but right now these regulations don't allow it, and certainly our politics would make it very tricky. But again, why are we sacrificing what could be important information to benefit live-born children and to benefit future fetuses that will become live-born children who could be given these therapies? Why are we sacrificing those possibilities in the name of the symbolic protections of a fetus that is going to be aborted anyway? This is deliberately provocative, but I did want to make sure that we at least put it on the table, assuming that there's some scientific value that could be achieved in that period of time. Consent is something that involves two questions: who gives the consent and what is necessary for the consent to be considered sufficient? Again, going back to this issue of direct benefit, if the benefit is solely to the fetus, in other words, the woman in theory has no self-interest in her own health in having this intervention, but the benefit is solely going to be directed to the fetus, then consent of the pregnant woman and of the father is required. There are exceptions if the father's unavailable or incompetent. There are exceptions if the pregnancy resulted from rape or incest, but otherwise, the father's consent is required. It does raise questions, though, about who is considered to be the father. Is it the male progenitor, or is it the intended rearing partner, assuming that there is one, which may be male or female or non-binary, because this can complicate what otherwise would seem to be a fairly straightforward requirement about who has to give the consent. As far as what the consent discussion should include, it has to include the variety of postnatal therapeutic interventions, as well as the possibility that even if successful, there might be miscarriage, stillbirth, or a child born with serious health problems, whether the problems you were trying to correct still present, although perhaps somewhat mediated, or a child born with serious health problems due to the intervention. I'd suggest that in addition to those kinds of things, there ought to be discussion about the local standards for how to manage severely impaired neonates. It's been my experience that from hospital to hospital and jurisdiction to jurisdiction, you have somewhat varied levels of deference to parental preferences about things like extraordinary measures for severely impaired neonates. There have been cases of tension between OBGYNs who have worked with the pregnant woman all along and maybe made plans in advance for what to do with a severely impaired neonate versus the neonatologists who would like to at least try to stabilize long enough for a fuller evaluation of the situation. It's just important that people understand what they're coming into so that they understand the range of decision-making available to them and factor that into whether they want to add another variable, this experimental intervention, into the situation. And then finally, whether abortion is available as an alternative. You know, the earlier that you can do these diagnoses, the more likely it is that somebody may still have abortion as an option. But that window is tightening up rapidly. We've seen in Texas, it's still under challenge in various ways, the effort to reduce that window to 6 weeks, which is, as you know, a time when many women are not even yet completely convinced they are pregnant. Mississippi now has a case going up to the Supreme Court that would place 15 weeks as the outside limit for abortion, other than for saving the life of the mother, maybe for her health. So we need to ask about the effect of these kinds of limitations. I was struck earlier about the survey about what people would do if they knew that they were carrying a child that would have these rather devastating problems. And that 60% of those who were surveyed said that they would not terminate the pregnancy. But those, if I read the slide correctly, were patients. In other words, people who'd already had experience with an affected child. And if that's the case, then their experience and their decisions may look very different from those of people who've yet to have had a child with these kinds of conditions and had the experience of trying to manage it postnatally. Many years ago, there was a survey that compared exactly these two populations, those with or without experience of a condition. And it was remarkable how differently they approach these things. And in general, how those who have had this experience tended simultaneously not to choose termination, but also to be very tolerant of those who would, whereas those without the experience tended to be very bifurcated into, of course I would terminate, and no one should ever terminate for any reason. Risk and benefit. Again, this came up a couple of times now. How should benefit and risk be measured? I would just want to highlight the fact that there's been an increasing level of discussion about whether disability in and of itself should be considered as a reduction in the quality of life. Surveys attempting to look at happiness levels among people with disabilities and those without, as well as kind of a diversity movement to think of disability as simply another variant. Because we're talking here about severe conditions, I think that this argument may not become that relevant, but as the range of conditions for which this kind of intervention may be considered increases, this discussion around disability and whether it should automatically be considered a disadvantage is going to become more salient. More close to this situation is whether the parental capacity to care for a disabled child is something that should be taken into account when doing this risk-benefit measurement. As opposed to looking at the risks and benefits just in terms of the medical situation of the child. Again, how should availability of abortion be factored into the calculus? If it is not an available option, it certainly does in some ways increase the need to try something that might result in a good outcome, but it also means that the risks associated with a failed intervention are even greater. Last, I'd just like to note that there is always the dilemma of parental disagreement. And in a situation with so many variables and that is so affected by general cultural and political attitudes about birth, women's obligations, abortion, and everything else, the possibility of parental disagreement is very, very real. And while in theory, each parent should have an equal say, often the experience has been that the person who says no is the one who really controls the outcome because there is such reluctance on the part of clinicians and hospitals to actively intervene against the wishes of either parent for fear of failure and further dispute. Last thing I'm going to highlight is simply the phenomenon of non-directive counseling, which has become quite a norm in many cases for genetic counseling. And certainly all options have to be discussed following the pregnancy with no intervention, abortion if it's still allowed, or doing the new fetal therapy, the intervention. The counseling is something where I would like to just emphasize some of the ways that counseling can be done. Obviously, information on outcomes for each option is where you start. But one of my colleagues at the University of Wisconsin, where I taught until recently, Gretchen Schwartzy, is a surgeon bioethicist. And Gretchen's done a lot of work on how to talk to people about surgical outcomes. And she found that simply giving people a description of an outcome and a percentage often didn't give them the same level of visceral appreciation of what they were discussing and choosing as a comparison of best and worst case scenarios. That seemed to most effectively get people to focus on what they were most frightened of and most hopeful for. And it led to a much better exploration of patient values, risk tolerance, and other aspects of the decision making. So this may be a slightly different way of approaching the counseling, where there's less focus on kind of numerical estimates and more focus really on what might your life look like if, or what might your child's life look like if this happened versus this happened, as a way to really explore patient values. With that, I'm going to turn it over to Charlotte Sumner from Hopkins to take on the next talk. Thanks very much.
Alta's talk was so phenomenal and really helpful in thinking about the ethical considerations of in utero gene targeting therapeutics. And today I hope to talk about a particular disease in which we've been really starting to think about moving forward to in utero therapeutics, a disease in which we've had real success in postnatal treatment, but there remains hope that perhaps in utero treatment could be even more successful. These are my disclosures. I do consult for a number of companies and receive funding from some of them. So the disease I want to talk to you about is this recessive motor neuron disease called spinal muscular atrophy, SMA. This has been introduced earlier today. This is considered a rare disease, but because its incidence is about 1 in 6 to 10,000 births, and the carrier frequency in the population is 1 in 40, it's actually historically been the leading inherited cause of infant mortality. It's a motor neuron disease, so motor neurons degenerate, and as a result, muscle becomes atrophied and very weak. And unfortunately, most patients have severe forms of the disease. This is a picture of an infant with type 1 disease. Again, historically, these infants develop weakness within weeks or months of birth. Because of profound weakness, never achieve the ability to sit independently, and without intervention, die before 2 years of life. There are also milder forms of the disease, what we call type 2 and type 3 SMA. A type 2 child is defined by one that does achieve the ability to sit, but never to stand. And a type 3 child develops the ability to stand, although usually loses this later in life. We also now understand that there are really extremes of the phenotype. There are infants that are rarely born with what we call type 0 SMA. These are infants that are weak at birth. And then a very mild form, type 4, that has adult onset disease. But again, unfortunately, the majority, 60% of patients, have this type 1 form of disease. So we've known the genetic cause of SMA since 1995, and the genetics of SMA have provided this really unique opportunity to develop various types of gene-targeted therapeutics. Most of us have an intact copy of the SMN1 gene, survival motor neuron 1 gene. This version of the gene always makes essentially a full-length mature mRNA transcript in a normal full-length protein. But patients with SMA have usually large homozygous deletion mutations of SMN1 and thus make no SMN protein from SMN1. But rely on retaining at least one copy of this sort of backup copy of the gene SMN2. Now, SMN2 is similar to SMN1, but has one nucleotide change that's particularly critical, a cytosine to thymine change within the exon 7 sequence of the gene. And that change sits within a splicing motif, such that when the pre-mRNAs from SMN2 are made, they are most often spliced to remove an exon, exon 7, and that shortened RNA makes a truncated protein that's rapidly degraded. But because of ongoing alternative splice events, sometimes exon 7 is retained and makes a full-length transcript and a full-length protein. The copy number of the SMN2 gene varies in individual patients. Those patients that have more severe forms of SMA have fewer copies of the SMN2 gene, whereas patients with milder forms of the disease have more copies, and there are some individuals with 5 copies of SMN2 who don't develop SMA. And this genetic observation was really the basis of remarkable progress in developing ways to treat this disease. And we're now in a place in the field where over the last 5 years, we've had 3 new treatments approved by the FDA. Each of these drugs works to increase SMN protein. There are 2 basic strategies to these drugs. One is to replace what's missing, the SMN1 gene, and this is done via an AAV9-based viral vector that packages the cDNA of SMN1. This is delivered IV and to infants 2 and less, produces more full-length protein, more full-length transcript in protein. The other basic strategy is to modify the splicing of the pre-mRNAs that arise from the SMN2 genes that are present in every patient with SMA. And treatment on this front has been successful in 2 basic ways. The first drug that was approved for SMA was nusinersen. This was approved in 2016. This is an antisense oligonucleotide. This drug binds the pre-mRNAs of SMN2 and acts as a steric hindrance to molecules that would remove exon 7 and thus promotes exon 7 retention and full-length protein production. The most recently approved drug, risdiplam, which was approved in just last year, is an orally bioavailable small molecule that achieves the same splicing effect, thus producing more SMN protein. So we're, again, in this remarkable place in the field where we have 3 different drugs. They're administered in 3 different ways. Risdiplam is orally bioavailable. It crosses the blood-brain barrier and has wide biodistribution. Omnisendergene abiprubart, the gene therapy, is currently administered only to young infants by a single IV injection. And nusinersen, the antisense oligonucleotide, is injected via lumbar intrathecal injection route. Now, each of these drugs has strengths and weaknesses. And, you know, it can be difficult now counseling families and patients about which drug to use. So that alone is complicated just with the postnatal choices of drug. But what is very clear for all of these drugs and all of the clinical trials that have been done is that earlier delivery of these drugs is much more efficacious. So when drug is given later in the disease course, there are fairly modest, if any, benefit of drug. But when the drug is given very early, either very early post birth or pre-symptomatically, there can be much more improvement in motor function. So just to emphasize this point, I wanted to show you some actual data. This is data from the ASO nusinersen clinical trials that were done by Ionis and Biogen. And you can see here on the y-axis a motor milestone score that maxes out at about 26 in infants. And you can see that unfortunately symptomatic sham-treated SMA infants with type 1 SMA never gain any motor milestones. Those infants with SMA that were symptomatic at the time that they received treatment had 2 copies of the SMN2 gene, so were severe SMA type 1 infants, but were symptomatic and less than 6 months of life when they received therapy had some improvement in their motor function. Certainly there was an improvement over sham, but what's so striking in this graphic is that those infants that were treated very early, less than 6 weeks of life, and importantly were asymptomatic at the time of treatment, have a much greater improvement in motor function. And those of you who are aware of the sort of miraculous improvements in SMA, we're talking about these kids that have this miraculous improvement, those that are treated very, very young. But even those infants who were treated very, very young with 2 copies of the SMN2 gene, again, this represents the majority of kids, still are not really achieving a cure. And that's important to remember. And certainly those infants that are treated post-symptomatically, you can see here at a year after treatment, only 8% were sitting independently, 49% had shown no improvement, and 16% had still died. So this just emphasizes the continued burden, particularly in infants treated post-symptomatically, but there is still, it is quite clear, there is not a complete cure, particularly with infants with 2 copies of SMA, even when delivered very quickly after birth. So, of course, these trial results have led to instituting newborn screening in the United States and in other parts of the world. This is an update about which states are now performing newborn screening for SMA, and this is rolling out across the country. What's been interesting about this experience to date, we had hoped, of course, that this would lead to earlier treatment, and of course it does. In general, treatment is initiated on average by about 3 weeks of life, and that actually seems pretty good, seems pretty fast, particularly for drugs that are complicated, extremely expensive. But unfortunately, the experience has been that those type 1 infants with 2 copies of SMN2, about half of those infants are becoming symptomatic before they're able to be treated. So why is this? So we've done some work looking at human tissues as well as studies in animal models to try and address why is there this really marked temporal dependency of the treatment. And one of the things that's clear is that if you look at the SMN protein, the protein that's deficient in this disease, it is not static during the course of development. This is looking at SMN protein levels in the human spinal cord collected at autopsy, at fetal cases, and in postnatal cases, either less than 3 months of life or older than that. And you can see here that the green are controls. You can see that the SMN levels are much more variable in the fetal period, but importantly, overall higher than they are postnatally. And there appears to be a decrease in SMN levels during this prenatal to postnatal period in particular. When you look at the SMA infants here in red, you can see that once you get out to these older time points, the difference between the SMA and controls is not statistically different. But if you look at this earlier neonatal period, and we only had a single case from the fetal period, but you can see that the difference there is greater, whereas these later postnatal periods, the difference is no longer statistically significant. So these data really argue that SMN levels are changing rapidly during development, and that SMA infants may be most deficient in SMN during in utero development. And that may be, in fact, when rescue would be most effective. We've also looked at the human pathology of this disease. This is a dissection that we've done at autopsy of a SMA type 1 infant. This is the ventral roots that contain the motor neuron axons and the dorsal roots. And you can see that in SMA children, these ventral roots are very, very small in caliber compared to a control. And those ventral roots contain a markedly reduced number of large myelinated axons. Motor neuron axons are large and myelinated in order to conduct quickly, and we can see that their number is very reduced in SMA children. So we wanted to understand why there was that reduction in those myelinated axons. And so what we did was we took this material to the EM, to electron microscopy, and what we saw actually is that many of these axons in the SMA human cases have a morphology that is resembling fetal nerve. So this is an example of what a fetal nerve looks like at 14 weeks of life. You see these tiny individual axons here that aren't yet separated from one another and aren't yet myelinated by a mature myelin sheath. And in the SMA autopsies, what we see is this huge overabundance of these immature axon morphologies, really indicating that SMA starts in utero. The failure of development of motor neurons is occurring very early and in fetal stages. To confirm that, we did longitudinal studies in mice, and we found that same pattern. That is that SMA motor axons in the mouse don't develop normally in utero. They're about the same in morphology at E13.5, but by E17.5, they're lagging in their normal development and they're too small. And then that period is followed neonatally by very rapid degeneration. And this, we think, because the axon isn't developing in the first place, makes it so vulnerable to very precipitous degeneration that occurs very quickly. So our model, when we think about SMA now, is that here are motor axons during fetal stages of development. They're very small in size, and they haven't yet developed a mature relationship with Schwann cells. Normally, Schwann cells should envelop individual motor axons and myelinate them. Unfortunately, in SMA infants, it appears that many of these axons have arrested development in utero, and that arrested development makes them very vulnerable to rapid degeneration postnatally. And if we miss this period of degeneration, this very precipitous degeneration, unfortunately, our interventions have minimal effect. And we think this is what explains why SMA infants, when they do well, have to be treated so early. This has been also observed with a biomarker that we can use in neurological disease. We know that when neurons die, they release a neuronal specific protein called neurofilament. And we've measured neurofilament in SMA mice. We can see that it's normal embryonically, but then in this postnatal days, day 1 and 2, it shoots up to very high levels and then sort of comes down again, highlighting this very rapid period of degeneration. And importantly, if you look in patients, you see that same phenomena. So here is data that was collected in collaboration with Biogen. You can see here that if you look at pre-symptomatic infants with 2 copies of SMN2, the dark pink here, so these are infants that are not yet showing any signs of SMA, but you can see that their neurofilament levels are a median of 33,000, whereas a non-SMA infant, their level is 167. And interestingly, as time goes on, the neurofilament levels are actually falling here in SMA patients, indicating that the most active disease is really at this very early neonatal stage. This is just, this phenomena is highlighted by this case that we had recently here at Johns Hopkins. It's a little girl who and her mother who consented to sharing their story. This is one of the many children we've taken care of who was identified recently by newborn screening, found to have 2 copies of SMN2, and was seen by our team that's led by Tom Crawford, who's a pediatric neuromuscular specialist, at 6 days of life. And she actually looked really good. At that stage, she essentially appeared normal. But when we went back and measured her neurofilament level, it was very elevated, even at this stage of life when she appeared clinically normal. Over the next 10 days, she very rapidly progressed. She developed very clear weakness of the trunk and the limbs, and her EMG was very abnormal, indicating, active denervation of muscle. She continued to get weaker for the next few days. Finally, all the things were put in place to allow treatment with gene therapy, omnisenogene avaparibec, which she got at 26 days of life. And as soon as she received the gene therapy, she did start to improve, and she's had slow improvement that one would never expect for a type 1 infant. She's had a slow gain of motor milestones. She's now 2 years old, and she can crawl and stand while being supported. And again, a type 1 infant would never normally achieve those milestones, but it's very clear that she still does have evident clinical weakness and that she will have, as Alta mentioned, disability for the rest of her life. And so I think, this question really highlights, if this little girl had been treated in utero, could she have actually been cured and never had to deal with weakness through the course of her life? And that's the big question. So can in utero treatment be more effective for severe SMA patients? We've been trying to answer this in preclinical models. We've been looking in SMA mice in particular at this point. The first experiment we tried was using a small molecule. This is similar to the risdiplam drug that I mentioned that's orally bioavailable and now approved for treatment of SMA patients. Because this drug has blood-brain barrier penetration, it also crosses the placenta. And so we delivered it to pregnant dams. And we could show that the earlier that we gave it embryonically to SMA mice and then continued it postnatally, the better outcomes we achieved. So this is just showing a motor function of writing time in mice. You can see that mice treated with drug just postnatally had impaired ability to write themselves in the first weeks of life. But when treated very early, these mice did much better. And when you look at the motor axons within the ventral roots, the earlier in utero treatment had significant improvement on the morphology of motor neuron axons. We're also exploring similar types of in utero therapy with splice switching ASOs, analogs of nusinersen. In this case, what we're doing is intraamniotic delivery. We're delivering at E13.5 to mice into the amniotic cavity. And what we see actually is mice that are treated embryonically, a single time embryonically, have even better weight and motor neuron numbers compared to mice that are treated postnatally. So we can see that with both of these two approaches, we get better outcomes if we deliver in utero than if we deliver just postnatally. And then finally, another group showed that in utero AAV9-based SMN gene therapy is also feasible in utero. They did this either by an ICV injection route or an IP injection route in utero, and showed that by both injection routes, they had improved survival relative to untreated mice. So, I think SMA provides a really important case example to think about key questions that need to be answered when thinking about whether we're going to go forward with in utero treatment. Is there a need for in utero treatment in SMA? There clearly is efficacious treatment postnatally, but there is also a suggestion that we could probably do better if we gave it earlier, but would the risk outweigh the benefit? Who should be treated? Should it be just the very, very severe patients or other patient types? When should treatment occur? Which drug should be used? How should it be administered? And what are the preclinical work that's necessary and needed in order to justify a human therapeutics development program? And we're particularly thinking about things about currently already approved drug versus a novel analog versus new routes of administration. Currently, for example, ASOs are being delivered by lumbar intrathecal administration, but in utero, we would probably do an ICV injection. And what does that mean in terms of the FDA approval process? I want to make one more point, which is that the SMA community does seem to be open to thinking about fetal therapy. This was a survey done by Marissa Schwab and Tippy McKenzie, together with the nonprofit foundation Cure SMA. And they asked the community, would you enroll in a phase 1 clinical trial of fetal ASO therapy or fetal gene therapy? And you can see that the majority of respondents would. And then would you choose to receive an FDA-approved ASO or fetal gene therapy? And you can see the overwhelming numbers who would be open to that. And then I'll just stop by thanking people in the lab whose work I showed you, in particular, Ling Kong, Dan Ramos, and my colleague, Tom Crawford, and numerous collaborators and funding organizations. Thank you. I am now going to introduce our next speaker, who is Ying Huang from the FDA.
Thanks, Dr. Sumner, for the introduction. Today, I will be presenting preclinical considerations for gene therapy products and FDA perspective. Next slide, please. In this talk, I will review the delivery of OTAT regulated products, describe a preclinical testing program in the 2013 FDA guidance for preclinical assessment of cell and gene therapy products, discuss animal species models consideration in preclinical assessments. I will have an in-depth discussion of safety assessment considerations for gene therapy products and those products incorporating the genome editing technology. I will then discuss early communication with CBER OTAT. Finally, I provide the selected FDA guidance documents. Next slide, please. OTAT regulates diversified gene therapy products as listed on the slide. The product examples that are the most relevant to today's talk include ex vivo genetically modified cells, non-viral and replication deficient viral vectors, typical adeno-associated viruses, AAV, and the genome editing products. Next, please. Preclinical assessment is an important component in the overall development pathway that a gene therapy product will follow from bench to bedside. The first FDA guidance on the preclinical assessment for cell and gene therapy products was published in 2013. It provides preclinical study considerations for objectives and the general program design. It provides recommendations for assessment of cell and gene therapy and the therapeutic vaccines to support an initial IND submission. It also provides recommendations for additional non-clinical testing to support an eventual BLA. It incorporates a strategy for 3 Rs, that is reduce, refine, replace animal use in preclinical testing. Importantly, the guidance specifies a science-driven review process to address safety issues in a context that considers both the biology of the product and the intended clinical indication. This approach incorporates the basic toxicological principles in preclinical testing while introducing a product-specific testing strategy. My talk is derived from the principle and the strategy of this guidance. Next, please. The 2013 guidance specifies preclinical program objectives, which should be addressed to inform the design of first-in-human study. The first objective is to support the rationale for the first-in-human study from preclinical proof of concept studies that describes the intended product for its biology activity and the pharmacologically effective dose range and the durability of these responses. An important note is that the cell and the gene therapy products are to be administered directly into disease subjects. Therefore, bioactivity of the product shown in appropriate animal models can help in interpreting the expected biological responsiveness in the intended disease population. The next objective is to demonstrate the safety for the administration to humans. This involves a comprehensive set of testing of toxicology, biodistribution, and other individual testing to permit identification, characterization, and quantification of potential local and systemic toxicities, their onset, that is acute or delayed, the possibility for resolution of any toxicities, and the effect of product dose level on toxicity findings. Next, please. For the third objective, the adequate of a dosing plan should be determined by preclinical data in animal models that address the risk to subject population and the clinical route of administration. For the next objective, preclinical data should inform the clinical trial design in selection of initial safe starting dose level, dose escalation scheme, and the dosing schedule. Preclinical data should also allow for identification of physiological parameters to inform clinical monitoring to mitigate the risk. Ultimately, the careful analysis will be incorporated in our review process to inform a balance of benefit versus risk profile in the context of the particular clinical indication under study. Next, please. This slide describes the types of preclinical testing for gene therapy products. Proof of concept study can be addressed using in vitro and animal models of disease to demonstrate the transgene expression and the bioactivity. We encourage assessment of biodistribution starting with proof of concept studies to gain idea for vector virus present in target and non-target tissues. Safety, that is toxicology and biodistribution, can be assessed in healthy animals to ensure sufficient study duration to be included. In some cases, safety data can also be obtained in animal models of disease to incorporate the POC, BD, and the toxicity. Next, please. Additional testing may be performed in a product-specific manner to address concerns related to tissue-specific toxicity, genotoxicity, tumorigenicity, germline transmission, or developmental reproductive toxicity. The testing may be incorporated in the definitive safety study. Next, please. Biodistribution assessment is an important component of safety evaluation. BD is defined as distribution, persistence, and clearance of the product in vivo from the site of administration to target and non-target tissues, including biofluids. BD assessments typically include analytical methods for the collected tissues and biofluid samples to detect the vector virus genomes, but may also include the methods to detect the expression product. BD can be assessed as a separate study or as a component of pharmacology toxicology study. Just to mention that the International Council for Harmonization has developed the S12 draft guideline on BD. The FDA cleared document of the S12 draft was posted in the Federal Register notice with the listed docket number. The public comment period is from September 9th to November 8th of the year, which is also posted on the ICH website if you are interested. Next slide. This slide provides considerations for selection for animal species models. This would involve use of non-standard test species in contrast to healthy animals that usually represent the standard model test system employed to conduct toxicology studies. For cell and gene therapy products, animal models of disease are frequently used to incorporate important parameters that allow for assessment of the bioactivity, biodistribution, and potential toxicity. Disease manifestations of animal models should be provided to allow for assessing clinically meaningful benefits and disease-related toxicity. Animal immune status is an important factor to be considered to allow for testing immunogenicity potential or allow for testing a human cell therapy products. Transgenic animals may also be used in some cases for testing a human cell therapy product. Oh, I'm sorry. transgenic animals may also be used in some cases. Other factors should also be considered. For example, permissiveness, susceptibility to infection by vectors, vector BD profile, and the transgene activity in the animals. Next slide, please. Now I'm discussing safety considerations for in vivo gene therapy products. The safety concerns for these products vary depending on the type of vector and the virus used, their biodistribution and the persistence in the non-targeted tissues and biofluids that is typically blood, and the level of viral replication in the body. Inappropriate immune activation by the vector virus transgenes is a major safety concern. Vector insertional mutagenesis and oncogenicity would be a concern for certain integrating vectors. Off-target effects and undesired on-target modifications from genome editing are significant safety concerns. Risks of the delivery procedure would be concerned for certain novel procedures. Next, please. Now I'm discussing safety concerns for ex vivo genetically modified cellular products. First, the potential toxicity is related to gene modification in cells, typically involving the vectors used in the genetic modification to introduce insertional mutagenesis and oncogenesis potential, and the other effects of off-target and unintended on-target modifications. The additional risk related to the administered cellular products can be the activation of undesired immune response to these cells, inappropriate cell proliferation or cell differentiation, cell migration to non-target areas, tissues resulting in undesired tissue cross-reactivity. For administration of allogeneic cells, graft-versus-host-disease is typically an adverse effect. Next, please. I'm now discussing genome editing technology considerations, as this technology has been applied to OTAT regulated gene therapy products. Current genome editing technologies commonly use engineered site-specific nucleases to introduce a double-stranded DNA break. There are 4 families of engineered nucleases: CRISPR-Cas systems, meganucleases, TALENs, and the zinc-finger nucleases. Base editor is an emerging genome editing technology that utilizes engineered nucleases and deaminase to enable the conversion of a specific DNA base into another at a targeted genomic locus. Next, please. Genome editing components may be delivered by viral vectors, plasmid, mRNA, protein, ribonucleotide proteins, INP, in order to process for genomic modification of cells ex vivo, or for targeted gene modification in vivo by direct administration of the engineered genome editing components. Next, please. Our preclinical considerations for the genome editing products are based on the risks in relationship to modifications of the genome. These modifications involve potential adverse effects due to genomic DNA cleavage at on and off-target sites. There might be off-target sites and events related to oncogene activation and the disruption of protein-encoding sequences and the gene regulatory elements, microRNAs, etc. Any effects of genome editing on chromosome integrity, including chromosome translocation and the genome rearrangement, will be the concern. The impact of on-target modification on the affected locus and the surrounding gene loci will also be among our considerations. Next, please. We also consider potential adverse effects due to gene mutation introduced by the nucleases and endogenous DNA repair activity. Immunogenicity is another safety consideration, given the foreign nature of genome editing components, overexpression of the transgene product that is nucleases, and undesired peptides proteins generated with genome editing. Adverse impact of the delivery system, such as a vector or lipid nanoparticles, can be additional considerations. Next, please. For preclinical assessment of genome editing product, we need the data to characterize the specificity and the frequency of non-targeting editing modification in relationship to the intended function and activity. For assessing off-target effects, multiple approaches are recommended. Initially, nomination of candidate off-target sites occurring in the genome should be using orthogonal approaches, typically in silico prediction using appropriate algorithm on the human genome database, a non-cell-based biochemical assay, cell-based assay, or whole genome sequencing. Importantly, appropriate assay thresholds are established, and the rationale for the selected threshold in appropriate biological systems for off-target nomination should be provided. Next, please. Subsequently, candidate off-target sites from nomination are analyzed to determine bona fide off-target sites. All candidate off-target sites nominated by the orthogonal approaches should be included in the analysis. For ex vivo gene therapy, the analysis should be conducted on the cellular product that is modified using the clinical manufacturing process. For in vivo gene therapy, the analysis should be conducted using the clinical product on vector, virus, target, and non-target cells, as well as in vivo system to assess the safety and tumorigenicity risk as appropriate. Next, please. Analysis of genome rearrangement should include a molecular analysis of the rearrangements within on-target sites and between on and off-target sites. Chromosomal structure analysis is needed to address any morphological changes associated with the genome editing. In addition, assays for analysis of larger deletions should be established to assist in the molecular analysis. Next, please. We also needed the data to support analysis of risk associated with off-target and undesired on-target effects. These can be evaluations for risks related to undesired fusion proteins and transcripts, disruption of coding genes and functional non-coding sequence, impact on chromosome integrity and stability, the evaluation for downstream effects such as cellular physiology, cell transformation, etc. Next, please. We do face challenges to addressing genomic editing safety concerns. For example, there are no standardized assays for identification of off-target genome modifications. Methods for evaluating large genome modifications or genomic instability are much needed. Use of various animal model species for safety evaluation may be limited due to significant difference in the genome between animals and humans. Genomic evaluation between individuals and the human can be accountable for safety-related outcomes. We encourage communication with OTAT at the early stage of the product development. The next 4 slides will give an overview of the process. First, about Interact. CBER implemented the Interact program in October 2018. The purpose of Interact is to provide a mechanism for early communication, which may benefit the investigator by non-binding, informal scientific discussion of specific issues with CBER review disciplines, That is CMC, PharmTox, and clinical. The timing is when a sponsor has generated the preliminary data for CMC and some safety data, but it's not yet ready to conduct definitive preclinical safety studies. Requests for interact meeting are made by email. Further details on the interact meeting process are described in the web link provided in the slide. Next, please. Although, along with the meeting request, the sponsor needs to provide a concise briefing package. The pharmacology toxicology information in briefing package contains the following: a comprehensive summary of all completed in vitro and in vivo evaluations that came from POC studies, preliminary safety studies, and the related references. Description of the preclinical development plan consists of completed and planned studies intended to support the rationale and safety of product administration in humans. Specific questions the sponsor would like to discuss regarding the submission should also be provided. Next, please. I also give some clarification about interact meetings. The meeting is not intended to discuss the designs for definitive safety studies, which are discussed in detail in the pre-IND meeting. It is not a forum to ask FDA to design a sponsor's preclinical program. Rather, FDA provides recommendations, feedback on a sponsor's plan program. Next, please. Pre-IND meeting is a formal meeting to communicate the initial development plan with the 3 review disciplines at CBER, especially CMC for product characterization issues, PharmTox for preclinical testing program, and for clinical, the scope and the design of a planned clinical trial. Therefore, a pre-IND meeting is held to facilitate an eventual IND submission. Next, please. In summary, comprehensive product characterization is key to understanding product risk. The preclinical testing program may need to be adapted to the specific gene therapy product and the level of perceived risk. New in vitro and in vivo test models should be considered as the science and technology advances. The 3 R's should be applied to preclinical testing programs. Communication with FDA at early stages of product development may be beneficial. Next, please. The next 2 slides list select FDA guidance documents for industry. I have discussed the 2013 preclinical guidance for cell gene therapy product. The 2015 guidance gives an overview approach to the design of early phase clinical trials of gene therapy products. The 2017 draft guidance describes various types of formal meetings, include pre-IND meetings. In 2020, FDA published 4 guidance documents for gene therapy products. The long-term follow-up guidance is one of them and includes recommendation of preclinical data of vector biodistribution, persistence, and integration, as well as an evaluation of products that involve genome editing for assessment of delayed risks in gene therapy clinical trials. Next, please. The other 3 guidance documents give considerations in clinical studies for 3 specific disease areas: hemophilia, rare diseases, and retinal disorders. Preclinical evaluation are provided with specific considerations for each of these disease areas. Next, please. This slide gives the contact information, including my email, if you have further questions about the presentation. Next, please. This concludes my presentation. Thank you for your attention. Our next speaker is Juan Gonzalez Velez from UCSF.
Thank you very much for the opportunity to speak with all of you today. I have no financial disclosures. We're going to talk about accessing the intravascular space for therapy. And in the literature, this is known as chordocentesis, or the alternative is PUBS, which is a percutaneous umbilical blood sample. The other alternative is to access the intrahepatic fetal vein. So the procedure has a long-standing history, initially described in the 1960s, 1964, with a hysterotomy, meaning an incision directly on the uterus to expose the umbilical cord and transfuse a baby that was hyperdropic. The procedure itself was successful, but unfortunately, the mother delivered prematurely and the neonate did not survive. There was a pause of other attempts until 1972, where an attempt was made with a cystoscope, pediatric in size, into the uterus to biopsy and to also sample vessels on the surface of the placenta for confirmation of the diagnosis of hemoglobinopathies. In 1974, the fetoscope was developed, and that led to a lot of successes in the 80s, doing both fetoscopic fetal blood sampling and fetoscopic intravascular transfusions for fetuses that were anemic. In 1983, with the advent and the improvement of ultrasound, this procedure was achieved under ultrasound guidance. And it is the method that we still perform today for chordocentesis. The indications include, the most common indication is fetuses that have severe fetal anemia, most commonly from alloimmunization. And we have a lot of experience with this procedure and a lot of safety data. The alternative, and in rare cases, there are situations where there could be fetal supraventricular tachycardia. We initiate treatment by medications like beta blockers to the mother with a hope that they transfer placentally to the fetus, but in some cases it's unsuccessful. So this also calls for a chordocentesis for fetal therapy. At UCSF, we have 2 clinical trials, the in utero stem cell transplantation for alpha thalassemia major, which has recruited multiple patients, and the in utero enzyme replacement therapy for lysosomal storage diseases. And both of these therapies, the cornerstone is chordocentesis. So how is the procedure performed? Usually the limitations are gestational age, and it's greater than 18 weeks. And this has to do more with a technical challenge. Below 18 weeks, the anatomical size of the vessels are too small to access safely. The 2 approaches are either the umbilical vein or the intrahepatic fetal vein. The preferable approach here in the United States is the umbilical vein, either a free loop or at the placental cord insertion. And Doppler is imperative when we do this to assure that we're accessing the vein and not inadvertently injuring the umbilical artery. As complications associated with the procedure significantly increase when the umbilical artery is injured. And these include fetal bradycardia from vasospasm, arterial spasm, and also bleeding from the vessel, which is under pressure. The team is a multidisciplinary team that we have. It includes the MFM specialists like myself, the sonography team, OB anesthesia, neonatology, and other colleagues that help us achieve this process safely. The procedure is usually performed in an operating room if the fetus is viable, meaning usually greater than 24 weeks, or if intervention is planned on behalf of the fetus if there is a rare complication. In pre-viable cases, it could be performed in a procedure room under sterile technique. The mother is placed in left lateral decubitus position just to improve circulation. Antenatal corticosteroids are administered in viable fetuses. Imaging is cornerstone, meaning ultrasound, and maternal intravenous access is usually achieved to deliver antibiotics for skin prophylaxis, anesthetics, and anxiolytics if they're needed for the mother. Tocolytics are used. Tocolytics are the medications to keep the uterus from contracting, and these include indomethacin or terbutaline. And as I mentioned before, both antibiotics and anesthesia are given to the mother. In some cases, fetal paralytic drugs are used. I usually do not. If the placenta is anterior, I reserve these only for the location of if the placenta is posterior to minimize fetal movement. And this is achieved with rocuronium. The needle that we use is called the Quincke point needle, tip needle. And this is either a 20 or a 22 gauge, depending on the gestational age. Less than 24 weeks, for sure, we'll be using a smaller needle, a 22 gauge. This is all the sterile equipment that we utilize. And as I mentioned earlier, ultrasound is key, especially a machine that is capable of performing Doppler to assure that we sample the vessel that we're going to enter and assure that we're in the umbilical vein. The machine is covered sterilely. Regarding complications, the biggest study is from the Netherlands, and our experience is similar to them. Any overall complication seems to be less than 1.2%. Preterm premature rupture of membranes is also very rare, less than 0.1%. Infection is very rare, 0.1%. Emergency cesarean section is less than 0.4%, and fetal loss is less than 0.6%. These complications are most commonly associated with non-immune hydrops if there's evidence of structural anomalies or severe fetal growth, intrauterine growth restriction. Many of these other complications like infection and fetal loss can be ameliorated with some of the other strategies that I mentioned earlier. Another key issue that has been demonstrated in the literature is that provider experience is cornerstone to decrease the complications associated with chordocentesis. And usually a provider is considered skilled with decreased complication rate when he or she reaches about 40 to 60 procedures. And to maintain competency, it's about 10 chordocentesis per year. Hence, this observation, most experts recommend that chordocentesis is limited to centers with adequate volume. So here I have on the slide, on the right side, we're demonstrating already the ultrasound probe sterilely covered using complete sterile technique. Usually the freehand technique is used to enter by the provider, him or herself, with a needle, and then once entry is achieved, the ultrasound probe is handed off to the sonographer, who will keep direct observation while the procedure is being performed. To assure stability of the needle is held obviously very carefully, and then we connect an extender, and through the extender we distribute either the drug or the blood or whatever is going to be administered. In this case, this is a demonstration from our in utero stem cell transplantation for alpha thalassemia major. And this is the actual ultrasound image from that video, and you can see how through the umbilical vein the therapy is being infused. So in conclusion, this is a very well-established procedure in maternal fetal medicine since the 1980s. It is safe at centers that have high volume to maintain operator experience, and it definitely has the future potential to deliver intravascular fetal therapies, including in utero gene therapy. So now I have the pleasure of introducing Dr. Nalan Gupta, who is professor at UCSF and is the chief of pediatric neurological surgery at our institution.
Thanks very much, Juan, and thank you to the organizers for inviting me to speak today. So the title of my presentation is Fetal Therapeutic Delivery to the Central Nervous System. And by way of background, as Juan alluded to, I'm one of the pediatric neurosurgeons at UCSF. I was the lead surgeon for the MOMS study, which evaluated fetal repair of myelomeningocele, and have experience with both cell-based therapeutics and gene therapy for a variety of disease conditions. I'll briefly touch on those before speaking about some of the specific considerations with fetal intervention. So the general problem technically, obviously, has been covered very well by the previous speakers, is the correction of gene defects in the central nervous system. And as was shown by Dr. Summers for SMN, theoretically, earlier intervention should, the goal is to improve outcomes in these patients that have progressive diseases. There are obviously many unanswered questions, and my goal is to show you that at least there's a path forward. In terms of classes of diseases, I think what most of us are speaking about right now are correction of single gene defects, which have a functional deficit that the goal of which we wish to repair and is measurable in terms of clinical trial settings. There are obviously other diseases that are more complex in terms of both their genotype and phenotype, those involving degenerative or irreversible conditions, and also those in the postnatal setting, particularly those that are neoplastic, so tumors and that arise in the nervous system. I'm going to just speak very briefly about a recent trial that we completed involving a group of children, not fetal, but young children who have a single gene defect that involves a failure of normal biosynthesis of dopamine or conversion of dopamine from precursors, and this is caused by the deficiency of L-amino acid decarboxylase. From a functional perspective, this is actually analogous to Parkinson's disease, except the mechanism of action, obviously in the children, is a gene defect, whereas in older patients with Parkinson's, it's a neurodegenerative condition involving those projection neurons that I'm going to speak about briefly. So the pathway actually, the gene or the enzyme in the center, the amino acid decarboxylase, actually metabolizes the production of both dopamine, noradrenaline, and adrenaline, and also serotonin. So children that lack that gene lack all of these downstream bioeffective molecules. We believe that even though the gene is, there's a germline defect in terms of the problem, the specific neuroanatomic structures that are affected are the projection neurons from the midbrain that do disperse to a variety of areas. The classical target on the left side is striatum, and it is these nigrostriatal pathways that degenerate in Parkinson's. It was, prior to this study, it was not clear whether these pathways are actually intact or are still present in children with the gene defect. So the advantages we have in the postnatal setting is that we can deliver, in this case, an AAV vector with very high precision in an MR environment to a target. And in this case, we perform the actual catheter placement and infusion under, this is a patient being treated in the bore of a 3T magnet in the operating room. And I'm just going to show just a very brief video. This is a sort of speeded up view of the infusion occurring in a patient in the substantia nigra on the right side. The reason you see it on MR is that we co-infuse gadolinium. The actual total volume of distribution is only 160 microliters, and we're covering about a total of about a cc of brain volume. This was recently reported, so I'm not going to go through the details of it. The main endpoints were motor function in terms of both what we call ocular gyre crises, which initially increase a little bit, but then diminish substantially. And then these are the individual subjects that were treated. both their ages at the time of treatment. The earliest was slightly over 4 years of age, and their improvement in motor function. There is a variability to their responses, but this GFM score on the left-hand side shows that the majority of them had very low function, but then did improve. So I don't have time to go through the sort of all the videos, and as I said, it's been published, but some of the patients who had never been ambulatory were able to walk, and a couple of them actually started to verbalize, which they had not done before. Through a variety of endpoints, we were able to demonstrate that the gene therapy vector was functional and resulted in an improvement in motor function in these children. How do we translate? So I guess my purpose of telling you about that is that we have the technical ability, at least in the postnatal setting, to deliver gene therapy, therapeutic agents to the central nervous system with high accuracy at very small and focal targets. In the fetal setting, we're limited clearly by our access to the fetus. The key questions are the anatomic target. Is it the entire brain or supratentorial compartment? Is it a focal target? And as Juan pointed out, the selection of route is critical. We have the most experience with intravascular. Intraparenchymal, and as I'm going to speak about in a minute, is technically probably the next most straightforward, and intraventricular for other variety of other reasons is also feasible, but more difficult. The last concern is the actual access route, whether it be through open hysterotomy or through an endoscopic fetoscopic procedure, or as Juan pointed out, an ultrasound-guided percutaneous technique. So in terms of preclinical studies, I was kindly asked by Tippy to join her and her group at UC Davis, where they have an excellent animal facility that has worked with a variety of clinical trials and uses a sheep model. This is an ultrasound of a fetal sheep brain, and I apologize, I don't have the calipers on here, but the ventricular system, which is here, is approximately 1 to 4 millimeters in size, so the target's very small. When we look at the cisterna magna in these same sheep, it's not much bigger, in this case about 3 or 4 millimeters in terms of target, but there's no bone overlying it, and we have a relatively straight route to it. I won't show that data, but we did an initial experiment, a set of experiments on a small number of animals looking at accessibility to the cisterna magna, ability to inject, and then also looking at dispersal of a labeled GFP vector to the sheep brain. And as I said, that's Tippy and her laboratory's work, so I won't provide that information except to tell you that it's at least technically feasible and demonstrates good distribution of the agent. There was a paper that was published just 2 or 3 years ago that just actually summarizes in this sense what was, they actually used this approach of placing bioglue in the cisterna magna of fetal sheep. Their goal was not therapeutic, it was actually to create a model of fetal hydrocephalus, and that was successful. But this is the exact same model that we would use in a large animal setting to test therapeutic agents. I'm just going to jump gears and say, well, is that feasible in human? This is a fetal MRI scan of a mid-gestation human fetus, and just happened to have this nice sort of sagittal image. The fetus in this case is obviously head upwards, but you can see in this case in the sagittal image, the cerebral cortex. Ventricular system is easily visible, but more importantly, the cisterna magna is also readily visible on this MR scan and also on ultrasound. We talk about direct ventricular or cisterna magna injection, and I think that is feasible. As you can see here, the occipital horn of the lateral ventricle is also relatively easily accessible. Obviously, the difference between cisterna magna and ventricle is that in this case, you're not traversing the brain in any way, whereas in this case, you are. Our experience with traversing the brain parenchyma, actually, we have a pretty good experience in preemies who have developed post-hemorrhagic hydrocephalus, and those children do tend to develop porencephalic cysts when we traverse the ventricular system. And so there's some theoretical disadvantage to a ventricular injection compared to one in the cisterna magna. I also draw your attention to the actual parenchyma of the brain, which is a target as well that could be utilized for very specific purposes. This is actually a twin gestation in a human fetus, and in this setting, I show this because one twin has a normal brain and the other twin has hydrocephalus. And in certain cases, hydrocephalic children have been already treated in a fetal setting to treat the hydrocephalus. And as you can see, technically, in a child with hydrocephalus, the ventricular system is greatly enlarged, but from a therapeutic perspective, this is very readily accessible. If you compare it to the normal brain, you can see that accessing the ventricular system, again, technically would be probably more feasible through the cisterna magna rather than the ventricle itself. The parenchyma is however, I think a readily accessible target in the future. There's also a lot of work being done in improving the MR imaging of the fetal brain. And then finally, I will say a couple of slides to conclude, which is that there is not nearly the extent of intravascular delivery, but in terms of accessing the accessing the ventricle to reduce fetal head size prior to delivery. And then also in a couple of groups where the ventricular system is accessed using a variety of fetoscopic tools to actually place ventricular amniotic shunts. In the United States, this has not been used or studied simply because we feel that the outcome in these children is already so poor. But this group in Poland and another group in Brazil have shown that this is feasible from a technical perspective. So in terms of conclusion, what I would tell you briefly is that access of the fetal CNS is technically feasible. The timeframe probably would be mid-gestation to the third trimester. The route of administration, meaning the selection of target, whether it be cisterna magna, ventricle, or parenchyma, is going to be a key factor in determining both the safety and risk profile, but also the tools required for the procedure. Thank you.
Okay, thank you very much for that presentation. I hope you can all see my slides. My name is Anna David. I'm an obstetrician and fetal medicine specialist at University College London. I'm going to be talking this evening about maternal and fetal considerations of prenatal somatic cell therapies. And my disclosures are here. I work for the Association of British Pharmaceutical Industry in a maternal health project, and I'm also consulting for CBER as part of a prenatal therapy for congenital skin disease. So we've been talking a lot about the fetus and thinking about the mother, but I put it to you that there are some other individuals who are, or potential individuals who are at risk. So clearly when we're treating the mother and the fetus, we've got both of those individuals who are at risk of having some kind of adverse event. But we also need to think about the fetus's children, so the mother's grandchildren, and this was very eloquently discussed by Chris Porada earlier on today. And we also need to think about the mother's subsequent children, because anything that might affect her ovary, for example, might affect her other children, but also potentially might prevent her from having further children as well. And, you know, what about the generation after that? Who knows whether our intervention, particularly if it has potential germline risks, might actually cause some problems. So this is a lot to think about. We also need to think about various aspects of the treatment. So it might be a drug, it might be CRISPR-Cas, it might be a surgery or catheter placement, stem cells, gene therapy, etc. So how do we think about this when we're doing clinical trials and interventions? We can think about it in the general health of the mother, the fetus, and obviously the neonate. And we also have some very, very important pregnancy-specific events which can be precipitated by our intervention, such as preterm birth, fetal growth, placental problems, accidents with the liquor, and also events around delivery. And I put it to you that these are extremely important, particularly for the mother. And she wants to know that the labour and delivery is going to be straightforward and that her fetus development isn't going to be compromised. When we're thinking about harm, we usually consider, from a regulatory perspective, adverse events. And the regulatory definition is any untoward medical occurrence in a patient or clinical trial participant who's given a medicinal product and which does not necessarily have a causal relationship with this product. So you might give a drug and it might not actually have caused this adverse event, but you don't know that until you've actually reviewed it and decided. And adverse events are really important because they are a fundamental part of clinical trial vocabulary. They're the language that we use to talk about safety in clinical trials. They facilitate swift and responsible communication of safety between study investigators, sponsors, and regulators. And regulatory guidelines require that they must be recorded in medical records. They have to be reported to the sponsor and the competent authority, and then determined if they're serious or related to the investigational medicinal product. But they're also used to develop clinical trial protocols, as I'm going to show you later on. And we look at adverse events, we determine their causality, whether they're unrelated, unlikely to be related, or possibly, probably, or definitely related to the intervention that we have. And the problem in pregnancy is there is a lack of standard grading criteria for adverse events for maternal and fetus. And so we've got a big issue if we're going to do really things like somatic gene therapy to the fetus. So I was working with a group developing a trial of an in utero treatment, a maternal gene therapy, giving the mother gene therapy to treat the fetus to try to improve fetal growth. And we were trying to think of how we're going to categorize safety in the mother and the fetus. So we went to the usual sources, the CTCAE, or Common Terminology Criteria for Adverse Events, developed by NIH, and we had a look at what there was available for adverse events. And there's 837 of them, but only 4 for pregnancy, puerperio, and the perinatal period, of which there was fetal death, preterm birth, fetal growth retardation, and a whole mix of other. Now that's just not going to be good enough. We will want to categorize fetal safety. So then we went to the Division of AIDS, which has done a lot of work, obviously developing vaccines and HIV treatment in pregnancy, and they list AEs with severity grading for common pregnancy complications. But again, these are mainly things like stillbirth, preterm birth, spontaneous miscarriage. So again, nothing particularly around the fetus. I had a look at the World Health Organization, and this classifies the severity of the preterm birth according to gestational age of delivery. So for example, extremely preterm, less than 28 weeks, or moderate late preterm after 32 weeks, but again, nothing specific for the fetus. And then we also had a look at the Clavian-Dindo classification for surgical complications, and that was more useful, in fact. There were grades all the way from grade 1 to 5, but they were specifically to do with interventions that were surgical, for example, related to endoscopic surgical radiological interventions. So again, not good enough for when we want to quantify adverse events in the fetus. Why does it matter? Well, it's very important for these phase 1 trials. It's going to be maternal safety that's key, fetal safety that's key. That's going to be our primary outcome measure. So we have to be able to measure it and to communicate it adequately. And we also might use safety to guide decisions in phase 1 trials. For example, we were talking earlier, there was a discussion about how do we dose escalate? Importantly, what are our stopping criteria? When do we stop a trial? Usually you might stop a trial when you get a grade 3 or grade 4 adverse reaction. But if we haven't got any definitions of adverse events to the fetus, then we've got a problem. And how do we compare adverse events in trial participants to adverse events in untreated women? We want to have a natural history or observational study to demonstrate that our treatment is going to not only improve the outcome for the fetus, but also be safe for the mother. And it's important for comparing safety between trials of different therapies or interventions. So it's really important that we have adverse event grading and definitions. There are some issues, though, about adverse events in pregnancy, because events can have a very different impact on the pregnant woman and the fetus. and I'll talk about that shortly. There may be a low threshold for admitting pregnant women for observation. For example, women will already be in hospital when they give birth. As an obstetrician myself, I'll just bring somebody in to monitor their blood pressure. That doesn't mean to say that it's an adverse event grade 3 or grade 4, but we need to define that. And it's also very difficult to assess the impact of an intervention on the fetus. We have cardiotoxicity, CTG or stress testing. We have some imaging, ultrasound, MRI. We can do fetal movement tracking, but there's not very much that we can really use. So taking this into consideration, and this trial that I was developing, we essentially developed a system which has now been published, and it's called MFET, Maternal Fetal Adverse Event Terminology. And here's the paper if you want to download it, and here's some resources that we've put online. And this was a labor of love that basically took us over 5 years to get to where we are. So we started off by looking at what was there existing in terms of grading criteria for adverse events. And we looked at national and international guidelines, as you can see, ACOG, RCOG, Australia, New Zealand, Society in Canada, ISPD, all the various guidelines, and we've identified what was available. We then convened a consensus group in Barcelona in May 2015, consisting of obstetricians, obstetric trialists, fetal medicine experts, fetal surgeons, pediatric surgeons, neonatologists, and industry representatives. And this was international. We had representatives from the US as well as Europe. And we came up with a draft set of 12 maternal and 19 fetal adverse event definitions and severity criteria. We then went to MEDRA, the Medical Dictionary of Regulatory Activities, and this was set up in the late 1990s by ICH to provide standardized medical terminology to facilitate sharing of regulatory information. And they basically took our new fetal adverse event terms. It was one of the largest collection of new terms that they'd ever had. And this was published in March 2016. So these are definitions of adverse events, but not the grading. We then had a discussion with charity representatives, patient participants, really thinking about how do they consider these adverse events? Are there any actual things that we've not considered properly? And this was very, very useful, and I think very pertinent to this audience. So for example, it will be important if a couple decide that they might want to have only palliative neonatal care after birth, or if they decide to terminate the pregnancy, this shouldn't be registered necessarily. It's a grade 5 adverse event, it's fetal death, but that's not the same as a spontaneous fetal death. They felt it was important to report mode of labour onset and mode of delivery, including whether the mode of delivery is likely to impact future pregnancies. They wanted it would be important to assess the psychological impact of the intervention on the pregnant woman, including the psychological impact on her of any fetal adverse events, and to evaluate it using validated measures. And where possible, to include assessment of the fetal response to an intervention, including indicators of fetal pain or stress. This is very difficult to do, but something that we should try to do. And then obviously to record data on subsequent fertility and pregnancies over a time period proportional relevant to the intervention. Are women trying to conceive, and are they being successful? So we then subjected our adverse event grading to Delphi consensus, 2 rounds with international involvement. As you can see here, this was the fetal adverse events. And we had clinicians, scientists, industry, midwifery, parent charity representatives. And this was the maternal adverse events. And I know a number of the people on the call very kindly contributed to this, and I'm incredibly grateful for that, because we couldn't have done it without you. Finally, we had consensus for almost all of the definitions, and we again met with the steering committee at a face-to-face meeting and came up with our final version. Now, this is version 1.0, because we do realise this is only a start. And if you're interested to contribute and you think we haven't considered something, please email me. I already have a few things that I need to add to version 1.1. So general principles. With a grading is based on the CTCAE generic criteria. This is here. So mild is grade 1, moderate, severe, or medically significant, including hospitalization, and then grade 5 is death. Now, as I said before, you can't hospitalize a fetus, so therefore we've taken that out. Grade 1 is mild, grade 3 is severe, requiring increased frequency of monitoring, and then fetal death is grade 5. And it's important that we've graded the severity independent for the pregnant woman and her fetus, because pregnancy conditions can affect the mother and the fetus separately. So for example, a woman might have very mild chorioamnionitis, a mild infection. It might lead her to deliver preterm, but then at 22 weeks of gestation, that might lead to fetal death, which is a grade 5. And hemorrhage in pregnancy might actually not compromise the fetus very much, but be significantly life-threatening for the mother. So this is our final version 1.0. As you can see at the top, we have maternal and fetal adverse events, and we have 4 that are the same, but affecting the mother and the fetus independently. We have hemorrhage, preterm premature rupture of the membranes, chorioamnionitis, and anemia of pregnancy. Then we have some fetal fluid collection. We have some fetal heart rate abnormalities, cardiac function abnormalities, some abnormalities of the fetal structures. So these would be new onset after the intervention was performed. And we have things like abnormal fetal growth, procedural hemorrhage, and post-procedural hemorrhage. So how might this manifest? Well, if we think about anemia in pregnancy, maternal, we have grade 1 up to grade 4, and grade 5 is always maternal or fetal death. So anemia in pregnancy might not affect the fetus very much in grade 1 to 3, but might be pathological, causing a pathological stress test, cardiotocography, indicating the fetus needs to be delivered. And looking more specifically at fetal adverse events, fetal fluid collection might be mild, grade 1, and then, but might be life-threatening grade 4. We've heard about hydrops, where we get fluid collection in at least 2 fetal compartments, which is sustained life-threatening, and it might lead to delivery, again, grade 5 being fetal death. And fetal cardiac abnormalities, we didn't feel there was much in grade 1 or 2, but grade 3 would be cardiac failure, and then grade 4 would be likely to lead to fetal injury or permanent disability. And it's also worth remembering there is a neonatal adverse event severity scale that's also been recently published in 2019, developed by the International Neonatal Consortium, and this gives you a comprehensive list of neonatal adverse events. So when we're thinking about putting together a trial and defining trial inclusion and exclusion, and particularly how to dose escalate, very often this is done to identify the maximum tolerated dose. And for the trial that I was doing, we were thinking about a trial of a novel therapeutic for early fetal growth restriction. You need to have enough pregnancies to be able to monitor the outcomes, but you also want to ensure that the neonate, that the fetus actually has severe fetal growth problems. And so when we're looking at clinical trials of new drugs, you can measure the safety by the occurrence of what's called dose-limiting toxicity events, which you define in the mother, the fetus, and the neonate. But what's the baseline dose-limiting toxicity? You need to know about the adverse events in the clinical trial eligible population. So in your observational study, you do observational study first, you find out what are your patients without your intervention, what are your fetuses without your intervention, how severe are they? And so we looked at this in women who had severe early onset fetal growth restriction, and we could identify mainly grade 3 adverse events in the mothers, and mainly grade 3 fetal adverse events, although we had a couple of grade 4 here. And in the neonatal adverse events as well, we had, for example, chronic lung disease grade 3, chronic lung disease grade 4, and dose-limiting events within 14 days of birth. And this basically allows us to decide which adverse events we're going to use for our dose-limiting toxicity. And because we had such a high rate of grade 3 in our natural history population, we've gone for dose-limiting toxicity of higher maternal grade 4 and 5, higher fetal grade 4 and 5, and also grade 4 neonatal adverse events. And so this allows us to say we're starting from a baseline of grade 3 adverse events. We expect to see those in our population. And so anything above that, grade 4 or 5, is going to be limiting our maximum, our ability to dose escalate. And we can set our target toxicity level as well. And this was done in conjunction with our clinical trials unit. So the future for this terminology is to disseminate promote the system so it's usable, working with regulatory authorities. As I said, it'll undergo revision. And the vision is to include it in all protocols of all trials in pregnancy. It's not just for clinical trials. You can use it to grade maternal and fetal adverse events in observational studies. You can use it to compare drug and surgical interventions. I've put a resources page on here, which gives you lots of information about the actual terminology, the grading, how we came about with it. And so in conclusion, it's a comprehensive, standardized system to define and grade maternal fetal adverse events. The definitions have been adopted by MEDRA. We used an international Delphi process to define and grade, and it should provide a meaningful understanding of safety and allow trials to be compared. If you want to know more about it, as there's our resources page, and we're very grateful to all the funders, the charities who worked with us at the European Commission, Wellcome Trust, NIHR, and our local charity as well, the EJ Hospital charity, that allowed us to deliver this after 6 years of a lot of work, and all of those on the Adverse Events Steering Committee. Thank you very much. And
thank you, Anna. That was really wonderful. I really enjoyed that we started with ethical considerations, ended with practical consideration of how we would consider adverse events, and then in the middle sort of issues of route of administration and practicalities of that sort. So it's a very interesting session. I remain a little bit confused, and I put this to everybody. I mean, Anna, you talked about a trial you're involved with in which the outcome was already very, very deleterious for the fetus. And so you start at this very high adverse event level, and therefore the intervention you can tolerate an even higher adverse event level. Whereas, in other disorders, perhaps the fetal manifestations are modest, but you're trying to optimize a treatment that already has some efficacy postnatally, but you might do even better if you shifted it to a prenatal timing. And yet the adverse events at baseline in that situation are very low. And so then your safety is going to have to be extraordinarily high. I wonder what each of the people on this panel would say about which kinds of diseases we should really be thinking about tackling first in utero scenario. These diseases which already have very adverse events in utero, but may be very difficult to show benefit in that case, or ones in which we have a clear, safe treatment that works postnatally, but then shifted to the prenatal period. I wonder if anybody has any thoughts.
I think the only example that I would give that I think has a interesting corollary to what you're mentioning is that the fetal interventions for fatal or fatal diseases was sort of the standard things like diaphragmatic hernia, twin transfusion. The fetal intervention for spina bifida or myelomeningocele was the first study in which there was acceptable postnatal treatment, but there was also pre-mom's data to suggest that there was a further additional benefit, but not without additional maternal risk. And so that ultimately was tested, which is rare unfortunately in our surgical world in a phase 3 type of randomized study. But I think as an illustrative case, it's an example of how, it is feasible to look at diseases that are non-fatal but have some serious morbidity. But then there has to be a clear intent that there will be an additional benefit to the fetus child that is balanced by, in this case, mainly the maternal risks that the mother had. So I think that balance is sometimes hard to achieve, and I think Alta mentioned that in terms of that balance of how do you calculate that? I think that's a difficult one. It has to be done disease by disease.
Anna and Alta, do you have thoughts from your perspective? Go ahead, Anna.
I was just going to say, I think you're right there. I think it has to be disease specific. And I think it also has to be related to the intervention that you're giving and how much information you know about it. So I think if it's if it's a protein therapy and it's something that is used and already licensed for use, and you know a lot about the safety, then I think you have more ability to say, well, probably, you know, the risk profile is relatively safe. It's given into the umbilical vein. We have a lot of information about that. We know what the maternal safety data on that is. So Juan mentioned the fetal safety data, but we've also published a systematic review of this maternal safety data of those kind of interventions. And actually, the outcomes are very good for the mother. So I think that's the sort of low-hanging fruit in many ways. You can go for that. And then probably step up through, you know, an ASO type intervention, and then more gene therapy, where you might be more worried about, you know, genome editing and that kind of thing. I think we have the ability now to take a stepwise approach rather than have to leap straight into gene therapy, which is a really difficult to know. There's lots of unknown unknowns.
So Ying, I mean, there are lots of questions I think that all of us have in terms of the regulatory framework for, you know, thinking about in utero gene targeting therapeutics. So I'm using that term very broadly. I realize gene therapy is very different from ASOs and so forth. But is there a specific regulatory framework that's already in place at the FDA specifically for in utero targeting therapeutics? And should there be, given their very special nature and the specific issues surrounding that? And I just wonder what your thoughts on that are.
Well, thank you for question. I think based on my understanding from the other conference, which is on the 22nd, I think FDA does not have the guidance specifically for fetal therapy. That's what I get from that workshop. However, I think in terms of if you consider gene therapy for preclinical evaluations, I think we do have a regulatory framework. For example, I mentioned as our 2013 preclinical guidance. Actually, that guidance covers a broad aspects of the gene therapy, including proof of concept, the safety study, etc. And they also has some discussion about the developmental and the reproductive toxicity. Although the timings, this type of toxicity would be different for the prenatal intervention as versus for the postnatal. I put that way, but at least the concept is there. It's about to test in developmental and the reproductive, which is both fetus and the mothers. I think that's, and also expanded from that part. FDA also has published, as I also listed their guidance on general concept of the clinical trial for rare disease. So that could be also applicable for fetal treatment, which is pretty much like you all defined as a rare disease. So that's one thing. This, I would think that's the framework, the baseline framework was there. Then also I would like to also extract to in terms of how we assessing the toxicity. There are some general principles from International Council of Harmonization, which is a famous S5 R3 guideline. Although this is particular for pharmaceutical, gene therapy is not in the scope, but the principle is applicable. And in addition, ICH consideration papers in general principles to address the risk of inadvertent germline integration of gene therapy vectors are more focused than the S5 on the gene therapy product for testing the inadvertent germline integration. So that I think could be also a useful document. And actually OTAT has experience in non-clinical assessment for DOT and for germline integration. Whether that could be used for the fetal therapy, I think that's different because like you all discussed, is different. There's a more complex than the postnatal administration. So in that sense, I would say, if you have a particular product in mind, I would encourage the sponsor, once they have the product in mind, that they decide the product and to consider earlier communication with OTAT for detailed guidance for type of assessment should be for this type of product. That is, I would say, was for the new product. I would say definitely is needed.
You know, the thing that's striking to me, you know, as in the neurological disease space, we've been putting these new drugs into the CSF space with very little understanding of what the PK/PD dynamics are of putting drugs into the CSF space. And, you know, providing the therapies to fetuses are sort of similar, right? We have limited understanding at this point of what the PK/PD dynamics are. We have to learn. I wonder if people have thoughts about whether, I mean, Ying, you emphasize the importance of biodistribution studies, but of course, the appropriate animal models for that in the fetal period remain really unsettled, I think. I mean, we assume that non-human primates would be the closest, but what about other large animal studies? That sheep have been, fetal lambs have been a model that have been used in this space. I wonder if people have comments about that, you know, the necessity of non-human primate studies for really assessing in utero, PK/PD, and biodistribution.
I think a lot of it depends on what you're putting in again. If you're putting in something which is potentially going to cross over to the mother, is going to cross the placenta, then you need to have more information about that placental transfer. Obviously, sort of large animals such as sheep are not really recognized, the data is not really recognized to GLP for regulators. So you're going to have to think about something like rats or rabbits, or even non-human primates. I mean, when we went with the work that I was doing about maternal gene therapy, we did approach a number of regulators, including MHRA and PI in Germany, and they didn't feel that non-human primates were going to be necessary because we could get the data from rats and rabbits, which were recognized animal models. But I think if you have concerns about the ability to transfer across the placenta, you can either do non-human primates or you can use placental perfusion models, so human placenta ex vivo perfused, which will last for about 5 hours, and that'll give you some information about whether a drug is going to cross over the placenta from the fetal side. You can infuse it on the fetal side and check the maternal, or you can check the maternal and infuse on the maternal side and check it on the fetal.
So
I would reinforce the. Oh,
go ahead, Noel, I'm sorry.
I think I would reinforce the fact that non-human primates, obviously a difficult model to work with in both non-fetal and fetal settings. However, the one issue is sense of scale in terms of the CNS. So if you're talking about delivery, so directly into the CNS, whether it be intracisternal, intraparenchymal, interventricular, it's hard really to study the adverse events of that in a rat or rabbit model. And I think some of the biological endpoints, pharmacokinetics, pharmacodynamics, can be done in that setting. I think the question is whether you can combine lamb, so lamb sheep as a large animal that reflects the size of the CNS, combined with a more feasible rodent rabbit model, and that's sufficient for the regulatory environment. Obviously, as I said, ideally non-human primate, but that it's a difficult model. We've done that for some of the gene therapy studies in the trial that I showed you, but those are expensive and difficult studies to do, clearly.
Ying, do you have any comment from the FDA perspective whether sheep would be acceptable to assess biodistribution?
Yeah, I think particularly for our gene therapy product, we do not require non-human primate as the default. We don't in general. And for this in utero administration fetal therapy, I would say provide evidence, provide the data to support your justification for the selection of animal species for study. Like the parameters you all discussed, cross-presenter concentration, and the target tissues in the fetuses. And I would add in that with the transduction of a gene therapy vector, the ability of gene therapy vector to transduce the tissue is another important parameter. And not necessarily non-human primates will be the best, but as long as you show the data to justify your selection of animal species that could support your study to determine the proof concept and the safety, that would be the way to go.
Anna, it sounds like you would like to answer Alta's question. Can you repeat the question for everybody?
Yeah, you put a question in the chat, which was a very important one. So the choice to hospitalize might vary by location, e.g. in low resource settings, people might not be hospitalized for situations that would lead high resource countries to admit to hospital. Absolutely. I noticed that one of the severity criteria 3, I think, referred to hospitalization. So would these criteria be setting specific? No, they wouldn't be setting specific. If you were running a trial, you could just choose that your grade for, that you would grade an adverse event as a 4 rather than a 3. So you might say that they would stay the same, but you just wouldn't choose to limit your study or stop your trial on the basis of a grade 4 where the mother might be hospitalized. Does that make sense? I mean, you know, this is up for discussion as well. This is only the first time it's been done, so there will be other things that will come into the mix. But as far as I'm aware, adverse event criteria, so CTCAE, etc., are not setting specific. They just apply across the board. But you can pick and choose what degree of grading you decide to use for your trial. So there is some flexibility around it.
Yeah, and it's just that most of your adverse events were measured by the physical condition of the fetus or the mother, but hospitalization is not about the condition, it's about the reaction. It's about the societal reaction to that condition, and that adds a new variable that is more complicated than the others. Well,
that's why you have to be so careful not to grade the fetus too severely. And that's why we took it completely out of the equation, that if the mother goes into hospital, that's irrelevant for the fetus. Because, you know, and then they might not be able to come into hospital conversely because it's 2-hour walk away or 1 day walk away or whatever. And so they actually don't go into hospital. Or they might be hospitalized for the treatment because actually in some low middle income countries, the hospital is so far away that they'd only get the intervention in the hospital. So that's why we actually split it. But it's an important point to consider.
We're running low on time, but I want to get in a couple other questions because I think this is such an interesting session. So Juan and Nalin, you each talked about the practicalities of actual delivery of some of these therapeutics that we've been discussing. Juan, you showed stats that I thought looked pretty convincing, that intravascular delivery is fairly safe, suggesting that at least at this stage, that would be overwhelmingly the preferred delivery route. Whereas Nalin, you mentioned feasibility, but really those delivery routes are much earlier in study, and so we don't really have these stats about really their safety. And in fact, it seemed to me in the setting of an infant that actually has a normal brain, that a delivery route to the ventricle where you would be crossing the parenchyma really would not be preferred. I just wonder each of your thoughts about this. I mean, obviously some of the therapeutics for neurological disease indications would be preferably delivered to the CNS directly rather than intravascularly. Do we have any hope of ever getting a CNS delivery fetally at a safety level that's acceptable?
I mean, I'll let Dr. Gupta, I mean, I think the safety around our procedure is just the historical perspective that we've been using it for alloimmune, and we have a lot of safety data around photosynthesis.
And I mean, the theoretical basis for doing something, directed to the CNS is exactly why we do it, I guess, postnatally, which is to reduce, systemic effects. So in other words, if we aren't at the stage yet where we have tissue specific, for example, therapeutic agents that are tissue specific. So, the holy grail would be a gene therapy vector that was expressed in a cell of interest that could be delivered by systemic route, because then you eliminate the risks of direct delivery and minimize the off-target effects. We're not there yet. So in some ways, I view the, whether it be cisterna magna or intraventricular delivery, I think you're right. Cisterna magna is probably, I think, in the human setting, far more feasible and less risky than intraventricular. But there are disease states. For example, when we treated postnatal kids with Pelizaeus-Merzbacher disease, which is a failure of myelination, that was a safety study. So we were treating restricted areas in the frontal lobe, but ideally you'd want to treat the entire brain. And the only way to do that right now, I think, would be intraparenchymal. But, I could envisage, more sophisticated vectors in the future that would eliminate that. Yeah.
Those vectors are coming fast and furious for sure. Okay, one last question. I'm gone over time, but I'll take my moderator's prerogative. Yang, I just want to ask you, so, when talking to industry partners about drugs that, they have currently, they've developed over decades and now have FDA approved, they're sometimes very resistant to thinking about trying to take that drug into the in utero setting, because if adverse events occur, then there's a worry that would impact their current label. Can you say anything about how the FDA would consider data that is acquired during a fetal trial and of a currently approved agent? How would the FDA consider that in terms of the safety of its postnatal use?
Thank you for question. Actually, for the labeling, I think I consulted our OTAT clinical lead on this. So I'm going to read to you what she sent to me about that labeling for the approved product for fetal therapy. I think what she said is regarding the labeling question for use of an appropriate gene therapy product in fetal therapy. I think what she said, while an approved product is investigated for an additional indication, in this case, it's like fetal therapy, the hypothetical product label is typically not updated. So you won't be able to update the label for the initial clinical trial. What gets updated is investigator's brochure for the investigational use of the product and informed consent. These two documents will be updated. When this your hypothetical program for new indication is complete, for example, you move into the later stage, or you think for the license, at that time there are two outcomes that could occur, according to her. One is that the product gets approved for the new indication and the product's label is then be updated with safety and efficacy data from your study. The other outcome would be when the new indication program failed and to demonstrate a benefit with the use of the product. In that case, it could be more complicated. The label may update the failed program contains data that are relevant to the safety and effective use of the product. And in order for safety considerations for the approved product use to be truly effective, that's I think the hypothetical new program has to generate sufficient data and produce a signal of serious risk that is reasonably attributed to the product, not to the indication, to the product. So this, I think, could be, according to her, could be difficult because postnatal use is different from prenatal use. How that signal could be reasonably attributed to the product is a question. That's what I get from her.
Yes. So what I took from that is it's complicated.
Right.
But there potentially could be some impact, which you know, it's a challenge because all of us here, I think, would like to see in utero therapeutics advanced, but that also obviously relies on close collaboration with industry partners. And so that does represent a concern for them. Okay, we're over, but thank you everybody for your talks and this panel. And I think we're gonna start again. We have a little bit of a break and we'll be back again at 1 o'clock. Thank you.